Welcome to AP Biology 2.7 Facilitated Diffusion. This is Dr. Joseph. This chapter is going to be a very short discussion. We are basically an extension of the previous chapter where we were looking at different types of transport, active and passive transport. In this chapter, we are going to look at how facilitated diffusion happens and the proteins that are involved in facilitated diffusion. That includes channel proteins and carrier proteins. And we are also going to look at subtypes of these channel proteins, uh, the ones that are gated and not gated, and what how they are different, right? So again, this is a quick recap from the last chapter. So diffusion is movement from the across a concentration gradient, and if you have small molecules, they are going to diffuse with no protein needed. But when you have large molecules but present in higher concentration on one side of the membrane and in lower concentration on the other side of the membrane, but are unable to move because they are either charged or they are too big to be transported through the membrane directly, then they take the help of proteins, in which case it is called as facilitated diffusion. And there are two types of proteins that are involved in facilitated diffusion. So here you have channel proteins. Channel proteins that are proteins that are basically opening up like tubes. Think of tubes, and then you have carrier proteins. Carrier proteins that change shape physically as they transport the molecule and carry it from one side to the other side. So here's again three-dimensional representation here. The main thing we need to remember is that channel proteins are going to transport ions and water, and carrier proteins are going to help transport molecules. So channel proteins work as pores, and the size of the pore determines what kind of molecules it is going to transport. And one of the things you'd see about channel proteins as well as carrier proteins is that because they are transmembrane proteins, the outside of these proteins are hydrophobic, and the inside usually acts as a hydrophilic transport area. So several things can cause the channel proteins to open or stay closed, and there are channel proteins that don't have any mechanism or gate to close or open. It stays always open. So that's what we see here in this figure. Here we are looking at transport of ions. In this case, we have an ion channel that is where when you have a neurotransmitter coming and binding to this channel protein, it causes the channel protein to open up and ions are transported, and then a binding of a receptor to this protein causes it to close and ions cannot move. They can also be open and closed because of mechanical pressure and also because of voltage. So accumulation of ions on one side of a membrane or protons on one side of a membrane is going to cause an electrochemical gradient, and that's going to become a driving force for molecules to move from one side to the other side. And the difference in that electrochemical gradient, the voltage, is going to cause the channels to open or then close. And here we have a non-gated channel, which is where which is just like an empty tube. But again, remember, being empty doesn't mean it's going to allow everything to pass. It still is very preferential about what it allows through, and it's unidirectional, and in some cases it is bidirectional. Just a closer view of how a closed channel opens up because of the binding of a ligand. So that is it. This is really simple, a very short chapter, and hopefully by now you're able to differentiate between simple diffusion and facilitated diffusion. And remember that facilitated diffusion is facilitated by channel proteins and carrier proteins, and the channel proteins can be either gated or not gated. And gated channels can be open because of voltage differences, or because of mechanical pressure, or because of binding of ligands. And carrier proteins are different from channel proteins in that they are able to physically carry materials from one side to the other side while undergoing physical change in the process. So that's the gist of chapter 2.7. Thank you.